The name of the principal investigator is Dr. Jerem Boeing. He is a senior research scientist at the CSR SARI, that is Savannah Agricultural Research Institute. He was employed in 2011, and he has a PhD in entomology from the Lincoln University, New Zealand. His research interest is in host plant resistance, insect ecology, habitat manipulation, agroecological pest management strategies, biological control of pests. He is the head of the Northern Region Farming Systems Research Group at the CSR Savannah Agricultural Research Institute. And he is also the chairman of the Editorial and Publication Committee at SARI. He is a principal investigator of two major projects at SARI. The first one is the pod borer resistant cowpea project, which is funded by the USAID through the African Agricultural Technology Foundation, AATF. And then he is also the principal investigator for Peanut Innovation Lab project, which is also funded by USAID through the North Carolina State University. So fellows and ladies and gentlemen gathered here, and those who are joining us by Zoom, I have the honor and the privilege to introduce to you our main speaker for this morning, Dr. Jerem Boeing. So, Dr. Jerry, you are welcome to GAS. On behalf of the acting president and the council and the Food and Agri chapter, I welcome you. So, without wasting time, I'll give you the floor to make the presentation for the fellows.
Thank you very much, Chairman, for the opportunity to make this presentation. And I'll just take this opportunity to thank the entire Academy for accepting to actually host us here. We've been doing a lot, and once we have eminent scientists such as you in the country, it is good we share what we have been doing with you so that at least once we update you, you can come up to speed with what we've been doing at the research level. So we do not take this particular opportunity lightly, but we very much appreciate it. And we'll do our very best to make sure that all the information that we need to share with you, we will share it with you. And whatever clarification that you will require, we'll do our very best to make sure that you have that clarification offered to you. What I'll be talking to you about today is the status of our pod borer resistant cowpea work. And this particular work is taking place at CSIR SARI. It started in 2011, and up to date, that's about 10 years down the line. So that's a lot of information out there. But I'm hoping that within this period, I'll be able to bring you up to speed with the most important issues as far as this work is concerned. So I'm the PI, but then I don't work alone, and I work with a team. So the next slide will show you the team. So that is myself up there as the PI. Then we have all these other guys there who do a lot of work. We have a multidisciplinary team made up of protectionists, that's entomologists. We have breeders. Then we have seed scientists, and also have technicians as part of the team. But then it's always good to acknowledge your predecessors. Dr. IDK Atopoulos started this pod borer resistant cowpea project work in SARI. He's currently on retirement. He was the first PI for this particular project. Then when he retired, Dr. Mumuni Abdullahi took over as the next PI. He retired in 2020. Then I took over from where Dr. Mumuni Abdullahi got to. The next slide, please. So briefly, to get everyone up to speed with what we are doing, we'll talk about the importance of cowpea and then the constraints in cowpea production, then the solutions that currently exist for cowpea farmers, then why we actually need GM as an alternative to pest management in cowpea. We also talk about what we have been doing at CSIR SARI as far as developing this BT cowpea that we call the pod borer resistant cowpea is concerned. Then the preparations we are making to actually have this variety released to farmers for cultivation. Next slide, please. So in terms of background, cowpea, usually we say cowpea, but then we are just talking about beans. It's nothing else but beans. That is what we call cowpea. It's an important source of plant protein. It has very good quality plant protein to make sure that human beings are able to meet part of their protein requirements. The other important thing about cowpea is that the leaves are also a good source of material for feeding our animals. So it's good fodder for our animals. And then, so basically all parts of the plants are important for taking care of, for meeting human needs in one way or the other. We focus a lot more on consuming the grains. Then animals feed on the hulls and other parts of it. Next slide, please. So what are some of the dishes that we get from cowpea? We can talk of kose, which most of us break our fast with it together with kuku. So once you have kose, you're able to break your fast. We talk about gari and beans, that's gobe, that people relish a lot, that is taken as lunch. We talk about our wache that is served at all kinds of occasions. Then in the northern part of the country, we have what we call tubani that is also made from beans or cowpea. And these meals are important sources of quality protein for human beings or for the Ghanaian population. But then in spite of the importance of this particular crop to our people, the major challenge is that on farmers' fields, farmers hardly get up to 1 ton per hectare when this particular crop is cultivated. On the average, we talk about 500 kilograms per hectare. That's what we record on farmers' field. But any farmer who is able to do a little bit more, the maximum the farmer gets to is about 1 ton per hectare, but it's not able to go beyond that. Meanwhile, if you look at most of the cowpea varieties that SARI has developed, or those that crop research has developed, normally the potential yields are 2 tons and above. So that means that farmers are losing over 1 ton. And there should be reasons why farmers are losing this particular 1 ton and not able to attain the potential yield of the crop. Now, it is common knowledge that the major reason why farmers lose this volume of their yield is the problem caused by insect pests on the crop. The important constraint to cowpea production on farmers' fields is the problem of insects. And these insects, they attack the crop at all stages of the crop. So right from the time the farmer plants, when the crop emerges from the soil, we have one group of insects that will come on the crop, then they will attack it. Then when the plant gets to the flowering stage, another group of insects comes on it, they also attack it and they cause all kinds of harm to it. Then once the flower, we have spores also coming up, another group of insects also come to attack it and cause all kinds of damage on the crop. So insects are the major limiting factor as far as cowpea production is concerned. And they are the reason why farmers are hardly able to record up to 1 ton per hectare on their field. Now let us look at some of the insects that attack the crop. The first and important one of the important ones that I'll talk about is what we call the thrips. Thrips are usually found in the flowers. So on my left-hand side, you can see pictures of the flower thrips there. Then in the middle, you can see a cowpea flower, and then on the extreme right, another cowpea flower. And you can see that these ones have been damaged by the thrips. So the thrips enter into the flowers, they feed on them, and once they start feeding inside the flowers, it causes what we call flower abscission or flower abortion. An important part of crop production is that if you want to have pod or you want to have yield, then definitely you will need the flowers, and it is these flowers that usually gives us the pods that we desire. So once thrips get in there and then the flowers drop, then that means that you cannot have pods. And if you cannot have pods, that means that the beans that we need for our gobe or whatever, we cannot have access to it. If thrips are uncontrolled, we get up to 100% yield loss in the crop. Apart from thrips, we also have another important group of insects that attack the crop. And all together, we call them the pod-sucking bugs. We have the giant curried bug, we have the clavigulera, and we have the legume pod borer, we have the sting bugs, the green and then the brown ones. all these also attack the crops, and mostly these ones will come on the crop when the pods are on. And once they attack the pods, like the name is suggesting, pod sucking. So they are sucking the green pods. And once they suck the green pods, that means that grain filling is highly affected and cannot take place. Sometimes in the course of punching the pod, they predispose the plant to what we call secondary pathogenic infection. So after they have punctured the pod and left, pathogens that are in the air take advantage of it, and then they colonize the entire structure. And once that happens, the pod gets starts deteriorating, and eventually the farmer cannot get anything meaningful from it. But then there is another important insect in cowpea production, which we call the legume pod borer. The scientific name is the Maruca vitrata. This particular insect, I refer to it as very important because unlike all the others that I talked about earlier, they attack at one particular developmental stage of the crop. But this one, it attacks it at the flowering stage and also at the podding stage. So while thrips are present at the flowering stage, this particular insect can also attack the flowers at that particular stage. And when it attacks the flower, you get the flowers eventually dropping or flower abscission also taking place. Then it also attacks it at the podding stage, also boring into the pods and feeding on the young developing grains. And when that also happens, that means that the farmer will not have any grain filling taking place and he will lose the crop. And because of that, he will not have any grains. Also, the holes that they make or the tunnels that they make through the pods expose the plant to secondary pathogenic infection. And this also results in the farmer losing the crop. So under heavy infestation, we've estimated that a farmer can lose close to 80% of the yield for that particular crop. An interesting thing about this particular insect, and one thing that makes it unique from all the others that I talked about earlier, is the fact that it has what we call a cryptic behavior. Once it is in the flower, a farmer can choose to apply insecticide. But because it is inside the flower, the insecticide is not able to effectively control it because it is protected by the floral parts of the flower. So what do farmers do? Currently, for this particular insect and for most other insects that attack the crop, we talk about for pest management, your first option is to use resistant varieties. As we talk now, as far as breeding programs across the African continent is concerned, and for cowpea in particular, we have been successful in identifying materials within the cowpea gene pool that are actually resistant to aphids. We also have materials that are promising for thrips. But then for Maruca, we don't have any material that we can use conventional breeding to actually develop resistant varieties for. So what that means is that whilst we are able to breed for resistance to thrips and then for aphids, we cannot do that for the legume pod borer or the Maruca. What this means is that farmers have to rely on insecticides to control this particular pest. Generally, on our farmers' field, farmers spray not less than 8 times just to control insects in general. But then the bulk of the spray application is aimed at targeting this particular insect that we call the legume pod borer. Because something like the pod sucking bugs that I showed you earlier, they are usually on the surface and they are huge. So once a farmer applies the insecticide once, he gets a high kill and then most of them are off. Others like aphids, if there's something like rainfall, even if you're not using an aphid resistant variety and there's rainfall, it washes them off. So the effect of the aphids is highly minimized. But then because this particular insect hides inside the flowers, the farmer has to repeatedly apply the insecticide just to control this particular pest. Now we know the dangers of insecticides in our farming system. The first one is the harm it causes to our environment. If a farmer applies insecticide and there's rain, it can easily be washed off into our water bodies, and that will kill other aquatic organisms that live in that are present in those water bodies. Even the human being that is applying the applicator is, if the person is not well dressed, the person may suffer the negative consequences of pesticide poisoning and other harms associated with using pesticides. Then for our farmers, most of these chemicals are very expensive. last year, some of the chemicals that we refer to as green chemicals, a liter was sold for about 46 last year. But as I talk to you this year, we are talking about close to 70 cedis for a liter of insecticide. Already our farmers are resourceful, so if they have to spend a lot more on insecticide just to control pests, that means that we are not actually helping them much. So the best way to sometimes manage some of these challenges is to look for approaches that are less expensive, but then they must be a lot more environmentally friendly. And that is where this particular project comes in, the pod borer resistant cowpea project. Basically, the overall goal is to contribute to food security and to improve the livelihood of our small-scale farmers in sub-Saharan Africa. And we try to do this by exploiting the power of biotechnology to develop improved high-yielding farmer-preferred varieties that are resistant to attack by Maruca vitrata, which in this particular case is the important insect pest in cowpea production. Now, why are we exploiting the power of biotechnology, or why are we talking of genetic modification for this particular insect? We are talking of genetic modification for this particular insect because there's been a lot of work that our breeders have done over the years. They screened not less than 15,000 cowpea accessions, both from the wild and from domesticated materials. And we've not been able to identify a single source of resistance or a single gene that can confer resistance to this particular insect in cowpea. So that means that within the cowpea gene pool, there is no resistance to this particular insect. So we cannot talk of developing a resistant variety using conventional plant breeding. Now, we found a closely related one relative of cowpea, that's Vigna vacillate, that has some form of resistance. But then crosses of this particular material with the cultivated one have so far proven unsuccessful, which means that we are back to the same point of not being able to use conventional breeding to actually come up with resistant cowpea varieties against this particular pest. So that is where we try to look at how we can exploit the power of biotechnology to use the BT technology to develop an improved variety. For decades, and in our organic farming systems, we rely on microbial pesticides that have Bacillus thuringiensis, which is a bacteria that is very common in our soils for pest management. And this particular bacteria, what it does is that in the course of its metabolism, it produces crystal proteins. And those proteins are what actually ends up killing the insect. So we have a lot of microbial pesticides made from this BT or Bacillus thuringiensis on our market. And we use it a lot in organic farming. So if I may digress a bit, if you look at our recent incident of fall armyworm in the country, one of the most recommended insecticide for fall armyworm control in the country was from BT. And it comes with the trade name Agro or Bipel. These are BT formulations. So we have these things that we've been using in our agriculture system for decades and globally. And that is where we identify our source of resistance from. Now, once BT is a bacteria in the soil, I also try to, though I'm not a plant breeder, but I'll try to talk a bit about the language of plant breeders. We have what they call, if you are trying to move a source of resistance from one organism into another, they call it transgenesis. Prof, I hope I'm right. It's transgenesis. So because Bacillus thuringiensis is a bacterium and we are moving it into cowpea, that's a form of transgenesis that we are trying to do. And to achieve that particular genetic modification, we went by what we call the Agrobacterium mediated transformation. So to get the BT gene from the Bacillus thuringiensis into our cowpea, we use what we call the Agrobacterium mediated transformation for the cowpea material. And the first cowpea material that we use is known as IT86D-1010. And this particular material is an IITA material. That means it was obtained from Africa here. It was subjected to that particular genetic transformation. The active ingredient that was used for the transformation to confer resistance against this particular insect is the Cry1AB protein. That is the protein that helps the plant to control this particular insect. Then, as part of the process, the markers, the selectable marker that was used to aid the process is the MPT2 protein. That is what was actually used to aid this whole transformation process. And that is basically how the genetic modification took place. So I'll just try to take a step back to just bring us up to speed with what there's processes of genetic modification. There are basically two ways of genetically modifying a crop. One is the Agrobacterium mediated transformation, which I described earlier. Then the second one is what we call the use of the gene bank. So A is the Agrobacterium mediated transformation, and that is what we use for the cowpea. Now, let me just explain further to say that for Agrobacterium mediated transformation, it even takes place naturally. So although we have GMOs that humans aid the process, but naturally too, we have GMOs in the system where the bacterium is present in the soil and it is helping to transform crops by transferring genes from one organism into crop. But just that this process is usually slower and it takes decades for that to actually occur. But then with the aid of biotechnology, we are able to take our understanding of what the bacteria does in the soil to actually modify our crops. So the bacteria assists to pick up the gene from Agrobacterium, helps to pick it up from the BT, and then it is put into the cell of the plant. Then through tissue culture, we go through the remaining process. And once we have a single plant or a single seed testing positive for the gene, you plant that one, then you multiply to get seeds from that particular one for the subsequent processes. The other approach, which is the B approach, where we use the gene gun, with that particular one, you bombard the cells of the plant with the particular gene that you want to introgress at a high frequency. And once it picks it up, you go through the rest of the process to pick a single seed to multiply to get the rest of the seeds that you need to continue with the rest of the processes. So for this particular project, it is important to always acknowledge your source of funding. What we are doing as far as the pod border resistant cowpea project is concerned is coordinated by a consortium known as the African Agricultural Technology Foundation, which has a headquarters in Nairobi, Kenya. The African Agricultural Technology Foundation is actually a group of eminent African scientists that have come together to form that consortium. So they put together proposals to funding agencies, and once they get the funds, technologies that will help our smallholder African farmers to improve their production are actually packaged and disseminated to them. For this particular project on the cowpea, it started as far back as 2004. But then after 2004, in Ghana and most participating countries, we didn't have our biosafety laws in place. We're still working on our biosafety laws. So the Commonwealth Scientific and Industrial Organization in Australia, they assisted us with the genetic transformation. So we provided the cowpea material from IITA, and then they helped us to do the genetic transformation. CSIR Savannah Agric Research Institute, we are another partner as far as this project is concerned, and we handle, once the transformation was done, we picked up the transformed material and we proceeded with the rest of the processes into coming out with a variety that is actually a GMO. This same project is implemented in Nigeria, and in Nigeria it is handled at the Ahmadu Bello University, the Institute of Agricultural Research. They handle this particular project. Then in Burkina Faso, INERA is actually handling it. And like I said, AATF is coordinating it, and the funding for this work is from the USAID. So I'll take you through what we have been doing at CSIR SARI. Basically, we've been doing a lot of testing and trying to develop this particular cowpea material. And why are we doing what we are doing at SARI? We are doing that because we want to make sure that whatever observation that we have from the lab can actually be transferred to the field. We don't want a situation where you will have lab results that are totally different from field results. We don't want to release the material to farmers and when the farmers plant, they see something totally different from what you told them they were supposed to see. So we confirm our lab observations on the field. Then we also do what we call efficacy of the gene under natural environmental condition. We want to be sure that once we have modified the crop, it is able to express itself under our ambient natural environmental condition. Then it is supposed to control a particular pest. We want to be sure that it does that. Then we have what the breeders call the genotype by environment interaction. We want to be sure that this G by E interaction, there wouldn't be any negative consequences in any way as a result of this kind of interaction. Now, whatever we are doing as SARI is highly regulated. We started with what used to be referred to as the National Biosafety Committee, which is now known as the National Biosafety Authority. And basically, they regulate everything that we do because Ghana is a signatory to the Cartagena Protocol. And once we sign on to that particular agreement, we have to put in place the necessary infrastructure to make sure that we can handle this kind of scientific research. So the NBA, they regulate and make sure that all what we are doing is done under confinement. Our genetic materials and everything is completely under confinement. And they ensure that whatever we say, the efficiency of the gene is actually what it is supposed to be. So this is how the front view of our confined field trial site. Like I said, GMO work is highly regulated in the country. You cannot just plant it outside. It is done in a fenced area and under 24/7 security. So this is the infrastructure that was put up at SARI for those works to actually take place. So the first planting of GMO in Ghana, or the first planting of GM cowpea in Ghana, was done on 19th September 2013. And the gentleman on my left is actually the first PI, that's Dr. Atokple. He was the first PI for that particular project. And the first planting he did, and that was in 19th September 2013. Then to my right, or the right of the screen, you can see a group of gentlemen there. That is the entomology group at SARI. So we had a number of materials and we were trying to subject them to heavy maruca infestation. Some of them were genetically modified. Others were just the normal conventional material. So they were subjected to heavy legume or maruca infestations just to see how they will react to the high pest pressure. So briefly, I'll take you through summary of what we've been doing since 2013 up to date. So the first set of transgenic materials that we evaluated as part of trying to develop our GM material, we looked at 7 materials. Out of the 7, the first one, that's IT86D-1010, was the material that was genetically modified to produce this number 2 to 7, that these 6 additional materials. So the first material is actually a non-transgenic material, but the other one, 709, 20, 1025, and all the others, they are all transgenic lines. So these were all transformed, and we needed to identify out of these 6 materials, which of them is highly efficacious against the insect that we actually try to target. So what we did was that we had those materials, all those 7 materials, the non-transgenic and the 6 other transgenic materials, they were planted in our normal 4 replicates. That was in 2013, and the work was repeated again in 2014. Then we reared the insect in the laboratory. So we raised a colony of the insect. Then every 3 days, between 5 and 6 a.m., we went to the field with first instar larvae of the insect. Then we made sure that at the flowering stage, each flower was subjected to heavy infestation of the maruca. So we put in about 40 larvae on each plant. And we repeated that consecutively every 3 days until the plant got to the mature, when it got to the podding stage and it was getting dry. That was when we stopped the infestation process. Now, basically, after doing that on all the 7 materials that I showed earlier, we realized that it was even 709A that was actually very, very effective in preventing damage by maruca. All the other lines that I showed, let me just back. So all the other life from number 3 to number 7, they were effective, but they were not as effective as 709A. So because of that, we selected event 709A as the most promising material, and we have an incinerator at the confined field trial site. So we burned the remaining materials because they were not supposed to be used for any other things once we identified a lead event. So once we identified 709A as the lead event, 709A is not a commercial cowpea variety in Ghana or in any African country. We used it as a donor material to now get the gene, that's the BT gene, into a commercially preferred variety. So like I mentioned earlier, it's 3 African countries that are participating in this particular work: Nigeria, Burkina Faso, and Ghana. To harmonize our data set for this particular work, we work with a variety that was released in Ghana by Sari Asongotra. And this material was released in 2008. And it's a common cowpea variety cultivated by our farmers. This same material was released in Nigeria and it was also released in Burkina Faso. So we now introgressed the gene from 709A into this particular commercial variety. And the picture down just shows how the introgression is done. So what the breeding team does is that they pick pollen from the 709A, then they transfer it into that of the Songotra once it is at the flowering stage. Now, once we got the gene into the 709A, we are normally able to test the material to be sure that you have the gene inside. And we do that using what we call some simple test strips that we use. And it tells us whether the process have been successful or not. So once we're able to develop a cowpea material that had the gene present, we needed to now subject that particular material to field testing. And to do that, we planted the cowpea under we planted it under a screen net condition, like you can see in the pictures there. Then when the plants got to the flowering stage, we repeated our efficacy test. So we read the maruca in the laboratory. Then we subjected each plant to at least 40 larvae during the flowering stage. And this was done at 3 days interval. So every 3 days, you've developed a BT cowpea material. You move in there to infest it with larvae of the maruca, and you do that every 3 days. When we did that, in the picture, that is the down picture, we have the non-BT up there and the BT down. So when we did that, we realized that the flowers for the BT Songotra were always remained and proceeded to the podding stage. But then the non-BT ones always lost the pods because the maruca destroyed the flowers, and then because of that, they couldn't have any pod coming up. At the yield stage, we realized that we're recording about 1.9 tons per hectare, which is actually the expected, which is nearer to the expected yield of the crop. That's the nearly 2 tons per hectare that the breeders normally provide as the potential yield of the crop. We got that when we had the BT, but then the non-BT we recorded below 1 ton per hectare. So what that basically meant was that the gene was actually effective at controlling the maruca. Once we had that particular result, we had to move on to do what we call the agronomic or phenotypic characterization. And what does this involve? This basically involves trying to demonstrate that the material that we have developed that has the BT gene in it is agronomically equivalent to the conventional cowpea or the non-BT cowpea, with the only difference being that it is able to resist damage by the insect that you are targeting. So the purpose of this agronomic trial was not to demonstrate the trait efficacy, but just to show that morphologically, the BT cowpea was the same as the non-BT cowpea. And to do this, we applied for permit to conduct multi-locational trials in our research field at Nyankpala, then Damango, and also at Manga around the Boku area. So we have 3 locations now for these agronomic trials. So for this particular agronomic trial, we had the cowpea material that was genetically modified, and then that's the Songotra that was genetically modified, and then the non-BT Songotra. And basically what we're looking out for was the plant height, the number of days to 50% flowering, number of pores per plant, then the number of maruca damage and all that. Basically, the results of this particular experiment showed that the BT was the same in terms of plant height, and then in terms of number of days to 50% flowering, in terms of then everything was the same as the non-BT one. The only difference that we recorded as far as this work was concerned was that the BT cowpea was able to resist damage by the legume pod borer, which was our target insect pest. So basically, the conclusion from that particular work was that there was no unintended effect as far as the transformation was concerned. Genetically transforming the cowpea did not result in any unintended effect. The growth of the plant was not affected. It was only the difference between the BT and non-BT was that the BT one was able to resist damage by the legume pod borer or the Maruca vitrata. Then the next issue was that we then needed to make sure that, well, we now have a material that is resistant to the insect that we are targeting. Is that material actually safe for humans? Is it safe for animals? That was the next set of questions that we moved on to answer. And to answer those particular questions, the first one is that after genetically modifying the crop, it is now producing a protein to protect itself against attack by the legume pod borer. Now, the first question is that, is the concentration of the protein, that's a cry protein, that the BT is producing in the plant, is it at a level that that when humans or other animals are exposed to, it will be harmful to them? That is the first level of safety that we were talking about. Then the second level of safety that we were trying to look at was that, is the concentration high enough to kill the insect, at least 99% of the insect, that's the LC99. Was the concentration high enough to actually kill 99% of susceptible insects? Because we needed to make sure that it's able to effectively control the legume pod borer at a high enough concentration, but then at the same time, it should not be harmful to humans or animals when they are exposed to it. So to answer that particular one, we took leaf samples, we took flower samples, we took pod samples, we took green cotyledons, we took dry seeds, we took pollen, anther, and then the roots. And basically what we did was that we analyzed for the quantities of the cry proteins, that is the protein that is giving the plant resistance to the legume pod borer. Then because the MPT2 was used as a selectable marker, we also needed to measure the amounts of that particular protein that was present in this particular plant parts. And those are the values that are provided in the table. Basically, for the cry proteins, what we realized was that the concentration was basically between 2.6 to 4.1 microgram per gram. And then for the MPT, it was about 1.5 microgram per gram. Now that was the concentration in there. But then too, we needed to know if this is the concentration in it, what will happen if livestock are exposed to this particular protein? Will, let's say, a cow, or will a sheep, or will any other poultry, whatever, when they feed on the grains or when they feed on the palms, will it be enough to actually cause harm to those particular animals? That's the daily dietary exposure to these particular proteins in the plant. So we did a rough calculation on it by making certain assumptions based on values that were obtained on the amount of feed these animals actually take. And basically using calculation on the concentrations of the proteins that using these particular averages to make those particular calculations, we realized that when it comes to the animals, there was no, if they were even to feed on only cowpea, for 365 days in a year, the amount of those proteins that they will consume was going to be 1.76 microgram body weight per milligram per kilogram of body weight. And that was far, far below what is high enough to actually cause any harm in those particular animals. We did similar estimates for human beings. Assuming the average Ghanaian decides to eat only cowpea every day for the 365 days of the year, we realize that the maximum amount of those proteins that the human being would have consumed is about 2.35 microgram per kilogram. And that is far, far below 400 milligram per kilogram body weight. That is stated as the maximum safety level for human beings to actually consume for this particular protein. The same thing for the MPT, where the maximum allowed concentration in human body is 5,000 milligram per kilogram. But then when we did the calculation of human beings being exposed to it for every single day, we got only 1.3 microgram, not even milligram per kilogram body, meaning that the concentration of this protein, whether in the diet of humans or even in that of animals, was far, far, far below what was lethal to any of these organisms. So basically, based on these calculations, what we simply concluded was that there was no way anybody could be exposed to high doses of these particular proteins expressed in the plant, such that the person will eventually suffer from any form of disorder because of consuming BT protein. The other fact is that this particular protein is specific. It even has nothing to do with human. It's specific to the epithelial cells of Lepidopteran insects and not just any other insect. So what it means is that if you have, let's take it that if you have Nizera, which is another insect, feeding on this one, it will have no effect on it because this particular protein is highly specific. It is targeting the epithelial cells in the gut of Lepidoptera, that is the caterpillars. They are the target. It doesn't affect any other organism, including human beings. Then bioinformatic analysis of the cry protein also shows that it easily breaks down. So once it gets into any living system, within a very short time, 90% of it is degraded within 2 minutes. So meaning that any non-target organism that this particular protein gets into, within 2 minutes, it is degraded. The same thing is that the cry protein is not toxic at dosages above up to 4,000 milligrams per kilogram. And the figures I showed earlier were even in micrograms, and they were below 2 micrograms per kilogram of body weight. So basically, based on the analysis that we did from the samples that were collected from our work, it shows that the cry proteins was not toxic to mammals and to any other organisms and even other insects apart from the Lepidoptera. And it didn't have any possibility of causing any form of allergy to humans or even to animals. Yeah. So once we had that particular result, the next thing was to do compositional analysis. And the aim of the compositional analysis was to find out whether nutrition-wise, the BT cowpea was similar to its non-BT counterpart. And the basic assumption for any compositional analysis is 2 things. One, you analyze the BT and the non-BT counterpart. And when you do those 2 analyses, you try to compare the amount of each nutrient that you have analyzed for in the BT and non-BT. If you compare and they are similar, and there's no significant variation between the BT and the non-BT, then that means that there's no concern. But then if you do that and then there's difference between the BT and non-BT in terms of the amount of the different nutrients that are present, you move on to compare that to what is expected in literature values, whether the values that you are seeing falls within that recommended in literature. And basically that was the foundation upon which we conducted our compositional analysis. But the interesting thing here was that when we did crude protein, crude fat, ash, carbohydrate, calories, and then the minerals, when we did analysis for all these in the BT and the non-BT one, The interesting thing was that there was no statistical difference between the BT and non-BT counterpart for these particular nutrient elements. So the crude protein in BT was the same as in the non-BT, fat was the same in BT and non-BT, and even the figures that we were recording for both BT and non-BT fell within that which was recorded in literature. So basically the conclusion here was that whether BT or non-BT, there was no significant difference between them in terms of the nutrient content. And this was done for samples collected from the 3 sites where these studies were conducted, that is Nyankpala, Damango, and then at Manga. Then we went on to analyze the leaf samples for the same nutrient elements. And basically it was the same results that we got. There was no difference between BT and non-BT for these particular nutrient elements. Then we looked at the mineral element, that's calcium, copper, zinc, manganese, magnesium, phosphorus, potassium, sodium, and then zinc. We looked at all those ones. The amounts of these ones in the BT and the non-BT one. And basically the result shows that there was no significant difference between BT and non-BT for this particular nutrient elements. The next important one is what we call the phytic acid. Phytic acids are they are actually anti-nutritional compounds. Normally, when they are present in a diet, they prevent the human body from absorbing important nutrients that the human body requires. So we analyze for the amount of these this phytic acid in both the BT and non-BT cowpea. And basically, the result that we got showed that whether BT or non-BT, the amount of these anti-nutritional elements were the same, and they didn't actually differ from what was actually reported of in the literature. So basically, from the combined site analysis that were done, the basic conclusion was that whether BT or non-BT, there was actually no difference when the compositional analysis were done, meaning that the BT one was equivalent to its non-BT counterpart, and that is actually what we normally see in this kind of way. The next thing was that, okay, so now we know that agronomically, our BT is the same as the non-BT, the conventional one. In terms of safety, we know that when it comes to the nutrition part, it is the same as the non-BT one. In terms of the toxin level, we know that there is no way that it has any lethal effect on any non-target organism, including human beings. Part of the effect on the environment, and that is where we went on to do additional analysis. So the first one was that we wanted to find out whether if we eventually release this material as a variety, has it got the potential of becoming a weed? This is important because if it becomes a weed, we all know the cost of invasive species, and no one wants to bring an invasive species into the country. So does it have that potential of one day becoming a weed? And to do that, we conducted a number of trials at our research station by using, and basically the outcome was that it was not possible. The reason being that cowpea, as we all know it has been domesticated, and the seeds of cowpea are so bulky that it cannot easily be moved from one place to the other. So that potential of it becoming a weed doesn't exist. It doesn't also have that long period of dormancy. Cowpea just has once there's moisture, it quickly begins to sprout and then to start producing. And because of that, that potential of it becoming a weed doesn't actually exist, whether BT or non-BT. Then the effect of pollen mediated, that's where the gene can move from, the BT gene can move into non-BT varieties. That is to other, whether outcrossing can actually occur when we have the BT and the non-BT one. We conducted studies on that particular one, and the body of literature available and supporting our work shows that there is, cowpea is generally a self-crossed material. So for pollen to move by wind, it is not possible. The only way pollen can move from cowpea to, from BT cowpea to non-BT cowpea will be through insects such as bees. It's only bees that can move pollen. And further studies that were done showed that the amount of such pollen transfer were generally less than 1% in all studies that were conducted, meaning that although outcrossing is possible, but because cowpea is self-pollinated, that cannot happen by wind. And if bees are to aid that particular process, the possibility is less than 1% of that occurrence, meaning that the potential impact of polymediated gene flow was very, very minimal. Then whether it could actually become a pest if it was cultivated, and that one too, the results show that no, like I said, cowpea, once it is domesticated, it doesn't have that hairiness to move about freely. And if you have cowpea, seeds dropping in a particular area, immediately there's a rainfall. There's no dormancy, prolonged dormancy period. It quickly sprouts, and once it sprouts, within a year, you will not find that same cowpea surviving there. So that potential of it actually becoming a weed pest is actually not there. We went on to look at the potential impact of the BT on non-target organisms. And for that particular one, we had a trial in this particular format. So we had a BT cowpea and non-BT cowpea planted. Then we put in yellow sticky traps, and we were monitoring the number of insects that visited the BT and then the non-BT cowpea. When we did that, we went ahead to actually count the number of insects that visited the BT and the non-BT field. The results basically show that whether BT or non-BT, the number of insect orders that visited it were actually not different. And what that basically meant was that the BT had no effect on insect visitation or other non-target organisms. Then once it had that, it didn't have any effect on other non-target organisms, we went on to look at the effect on alternate hosts. Now, we needed to be sure that if the BT is to be cultivated in a very large area, the next question that we sought answers for was that if Maruca cannot survive on the BT, what other crops can they survive on? So we need to find out whether they have alternative hosts, because the aim of this project is not to wipe out Maruca from the ecosystem, but then we wanted to make sure they have other alternative sources of surviving in the environment. And to do this, within our confined field trial area, we scouted 200 to 300 meters away and tried to identify other potential sources of alternative hosts for this particular insect. And basically, when we did that, we're trying to identify what plants can Maruca survive on. And if they can survive on, we wanted to actually find the Maruca inside the flower bud, or wanted to find symptoms of Maruca damage on flowers of those particular plants within the vicinity. So after doing that, we identified about 9 different plant species that Maruca can survive on within our environment. And these plants were found within a 300-meter periphery of the site where these trials were conducted. One was the Tephrosia species. We identified Clotorrhea as another alternative host. Senna was identified, Cocorus was identified, Styloxanthus was identified, Fururia was identified. So the conclusion was that what this basically meant was that the insect that we are targeting, in the absence of cowpea, there were other crops that they could actually survive on, which meant that they had alternative foods that they could survive on, and they could continue to reproduce in those particular crops. Then the other one was that how many sprays will a farmer need to spray his BT cowpea field? I said earlier on that cowpea has a number of insects attacking it. It's not just Maruca. So how many sprays will a farmer need to apply to target the other insects that the BT trait does not control? And we conducted an experiment where a replicated experiment where the crop was either the BT and BT cowpea was not sprayed. Another set was sprayed twice, another set was sprayed 5 times. It was a split crop experimental design. And the results basically showed that the BT always yielded higher, whether under no spray condition, under 2 spray or under 5 spray condition. BT always yielded higher than the non-BT. And that was because of effective Maruca control in it. Then we realized that whether you sprayed 2 times or 5 times, there was no significant difference between BT and non-BT for yield. And what that basically meant was that farmers who decide to crop BT cowpea do not need to spray more than 5 times to attain the maximum yields that are possible. But then these days, you don't just develop a material, but when you develop a material, you must work with farmers. So still within our confined field trial size, at Nyankwala Damangu and Manga, we invited farmers to come and assist us to actually evaluate the materials. So they were given seeds that were just labeled A, B, C, but they were not told which seed was which one. And they were allowed to plant it using their own farmer practice. At the end of it, we did a small survey to find out from them which of the materials they preferred. And the simple summary of it was that all farmers expressed interest in the BT because they realized that they had more yield, and that's what our farmers are always interested in, yield. They had more yield from the BT than the non-BT one. So what that basically meant was that farmers also showed a lot of interest in this particular material. And in preparation to make sure that we can have this material available to farmers, we have produced about a ton of breeder seed that we can move into multi-locational trials if the National Biosafety Authority grants us a permit to release this material as a variety. But then in the short term, all what I've been talking about is just a single cowpea variety that we have worked on. We are working on actually having this gene available in other commercial cowpea varieties that our farmers are growing. So we have Cake House Benga, we have Padituya, we have Wanka, we have Apabala. These are other varieties that our farmers prefer. So we want to make sure that we have this gene in these other varieties so that a farmer can have choice as to which particular variety he wants and not just be stuck to a single variety. And once we have that, what it means is that farmers will save costs because they will no longer have to spray excessively to control Maruca on their farm. It will increase their income and also preserve our environment. As part of strategies to prevent insect resistance, we have a plan B for insect resistance management. As I talk to you now, we are almost concluding work on a second gene, Cry2AB gene, that will later introduce as a backup gene to further strengthen the resistance in the cowpea. And that is just to make sure that farmers will not plant it and then maybe years to come, the resistance will break down. As part of all what we have been doing, we've been engaging a lot of stakeholders. We had a number of audience with the Agric Minister as far back as 2020, trying to engage him on our work. We had another meeting last year with the Minister for Environment, Science, Technology and Innovation. That's our sector ministry. We've been engaging from Ministry of Agric Workers at the district level. That's the District Agri Development Unit, the Regional Agri Development Unit. We've been engaging media. We've engaged seed producers. We have an institutional biosafety committee that we engage in all that we have been doing. So these are a few of our engagements with farmers for them to see how the material looks like and to show their preference. These are meetings with Agric staff at the institute and also with media and other people just to bring them up to speed with what we are doing. The first picture was at Nyankwala. This is at Manga in the Upper East region where we try to interact with our collaborators. Our institutional biosafety committee has oversight responsibility over what we are doing. And this is just one of their meetings to review our activities as far as this work is concerned. Then somewhere last year, we were able to completely put together a document to apply for environmental relief to the National Biosafety Authority. As I mentioned earlier, all this work we've been doing has been under confinement. So we applied to the National Biosafety Authority for environmental release. Environmental release simply means that for us to work outside confinement so that eventually we can work with the varietal release committee to have this material out as a variety. So we submitted our application to them last year. Our Director General, our former outgone Director General, he did the submission on our behalf to the National Biosafety Authority at Kwabenya. Then since it was submitted, the National Biosafety Authority have gone ahead to gazette the application, seeking for public comment or public input into the application. That period elapsed. It was a 60-day period and it has elapsed. It elapsed somewhere in April. Then I think the Technical Advisory Committee of the National Biosafety Authority, they met on the 16th to 17th of May, that's of this month, to actually conduct a risk assessment on our application. So basically, that is what we've been doing as far as trying to get the the first genetically modified cowpea material in the environment and trying to answer all the questions right from agronomic to safety issues as far as having a BT cowpea in the country is concerned. Thank you
very much, Jerry. I think he has really given us something for discussion. He has walked us through the entire process, and I will now want to open the floor to fellows, those of us here and those who are joining us by Zoom. If you have any question or comments, Jerry is here to respond to any question you have concerning the BT cowpea. So the floor is now open. Propriety. You're an entomologist. Do you have any questions?
I'm impressed with the presentation. I would wish that this presentation is made in a form that many more people can get copies, especially the process that they've gone through. So the people who are against GM need to be educated to understand the process. The process is very important so that they are here to be allayed. And so I would suggest that you find a way of publicizing this more effectively.
Because you're an entomologist and you appreciate the entire process, yes, that it is based on science,
yes,
and they have data to support work that has been done. So the floor is still open for comments and questions.
Yeah, so greetings from Tamale. I'm visiting Tamale. Yes, thanks very much for a very impressive presentation. This shows the extent of work done, which is so impressive. One or two comments. Jerry, your presentation, you were talking about spraying 5 times versus forgotten what. But most of the time, we credit this thing with farmers spraying up to 8 times versus 2 times only on BT. That's often cited in the literature in terms of farmer practice. The other area I want you to comment on is the stewardship of this particular material. If it is eventually released to farmers and they don't handle it well, the insects may become resistant by mutation, and that's a natural process. I wonder whether your interaction with farmers will also hammer on that. How do you avoid this buildup? You mentioned other plants where the insects can move into, and that also will reduce the tendency to mutate, become resistant. But I wish you would hammer on these stewardship aspects when you release the material. Or maybe you are waiting for the environmental permit to be released before you can then interact with farmers. But even ab initio, I think you should start hinting that these insects can become resistant. That's why what you've done. And you talked about resistance management to benefit the farmers. I think that's all I have to say for now.
Please, we're going to take 2 more questions in addition to what Prof. Alaza has asked, and then we'll give the floor to Jerry to respond. So,
thank you very much. I've joined only about 15 minutes ago. I was in traffic and I had to turn around. So I missed the physical and only got the tail end of it. Very brilliant presentation from the little that I've seen. Let me ask questions that some people have been asking me. Hey, you people, you are introducing this, that sort of thing. You did a crude protein analysis and you found no difference between the BT and non-BT. If my biochemistry is limited, I'm the main population geneticist. But if I remember what I was taught, crude protein, you are essentially measuring what? Is it a nitrogen and other things? So it may well be that the crude proteins may be different or may be the same, but the components, is it okay to go further and identify the actual proteins that are there so that we are sure that they are not poisonous material? 2 materials may have the same crude protein content, but one can easily be a deadly poison because of the configuration. Can you bring in some X-ray crystallographers and whatnot to identify these proteins and make sure that, yes, these are good ones? Because if you start eating the BT today, somebody will say that cumulatively over the years, it may, during your old age, perhaps it may have accumulated certain things in your body. I'm asking this question not only for myself, but because I haven't been able to get answers to some of the questions that I have been asked. So, Jerry, please help me out.
Jerry, thank you for an excellent presentation. But I'd like to ask a few questions. When you compare your BT cowpea and the normal cowpea, what is the ability of the BT to use nitrogen from fertilizer or manure, comparing the two? And then secondly, you talked about the fact that there was no problem. I mean, you gave it to the farmers, they grew, I mean, they planted it and it came up again. Was it just one time or more than, one time? So, and the last question I want to ask is that, the chairman has worked on cowpea for some time. Jerry, does it mean that your BT cowpea is far, far superior than our normal cowpea, or there must be some disadvantage that it has, or something that our normal cowpea has over the BT cowpea? Does it mean that everything is wonderful about the BT cowpea and is far superior to our normal cowpea? Because I know the chairman has worked on cowpea for a very long time. So one, what is his ability to use nitrogen from fertilizer or manure? Two, you gave it to the farmers, they planted it, it came up very nicely. Once, was it once, twice, three times, And the last thing is comparing it to what we know. Is there any disadvantage on the BT cowpea? Thank you. But well done. Thank you.
From Professor Dampton. But you did not test the new breed against any of the pre- and post-emergent fungal diseases. Jerry, the opportunity to respond to these three questions from our fellows.
Okay, thank you very much. I think it's fair to start from the first question that came from Prof. Walter Alassan. So yes, he was asking about 2 sprays and then the multiple sprays. So basically, when we talk of the BT cowpea or the resistant cowpea, currently what farmers do is that they spray not less than 8 times in the course of the season. And even when we talk of seed producers, they even spray sometimes up to 10 times. And if you talk to farmers on their field, some will even tell you that every 3 days they apply insecticides on their farm just to control insect pests. But what we are saying is that we tested up to 5 times. And even up to the 5 times, what we realized was that the BT required not more than 2 sprays. That was all what the BT required. So if a farmer is cultivating this particular variety, he doesn't need to get up to 5 or 8 or 10 sprays in the course of the season. He just needs 2 sprays, one at the flower bud initiation to take care of a few trips that may be there. Then the second one to take care of pot sucking bugs. Those are the main sprays that are applied, and that's what it targets. But the remaining sprays that usually targets the maruca, the farmer doesn't need to apply that. And those are the ones that pushes the number of sprays to about 8 or 10 because of the cryptic behavior of the insect, because it hides inside the flowers and the pods. So that's why they do repeated application against these particular ones. The issue of stewardship, yes, Prof, I think you are right. One thing about BT varieties is that it goes with a lot of stewardship issues. And stewardship basically means that getting the farmers up to speed with how to handle the technology. And once you have good stewardship, you are able to delay resistance development. I didn't talk so much about stewardship, but stewardship is part of our plan. In fact, we will set up, if we actually have the variety coming up, we will set up a National Stewardship Management Committee to assist in the process. Even at the regional level, we'll have a stewardship committee. And basically what they will be doing, it will be made up of people from the agri development units and even from agri extension directorate too. And what they will be doing basically is to help us to monitor, so that if suddenly any farmer says that I planted BT and saw this, then quickly a report can be made and we can quickly follow up to actually confirm what the farmer is saying, to know whether it is true or not, and to identify where the problem is, so that we can quickly tackle it. So stewardship is an important part of this process, and then it's part of the plans. We'll set up the national stewardship and also the regional level to handle some of these things when the variety eventually comes up. And there'll be a number of workshops and training to build the capacity of those who be part of the stewardship management so that they can be up to speed with it. Then the next set of questions were on the crude protein. The crude protein, because of protein is made up of different elements, and the way they are arranged can either make it deadly or non-deadly. Fortunately, this concern is actually genuine, because it is one of the issues that one of our opponents actually raised. But interestingly, Prof, I can assure you, if you look at the way the genetic transformation was done, it adequately handles this particular issue, because in the slide that I showed, where I showed the circle with how the transformation was done, it's just a single insert, and it adequately handles this particular issue of the protein. So the protein that we measured in the BT was not any different from what was measured in the non-BT in terms of the constituent. We actually have a comprehensive document that has all this information in there. And I think for now, I'll say it is almost a public document that can be accessed from the Biosafety Authority. Let me see if I can get back to that slide. I can move around. So I'm just trying, this is the diagram I'm talking about. So this is about the way the genetic transformation was done. So the insertion was so precise, such that whatever we are seeing in terms of the expression of the proteins will not have any kind of negative effect in any way. So what we see in the BT and the non-BT, we saying that they are the same actually means that they are the same. There wouldn't be any form of maybe the high amounts of lysine in one place and maybe low amount of another component in a different place. No, the composition will basically be the same because of the way the genetic transformation was actually done. So that particular fear, I don't think it actually exists. Then the other one,
I didn't see this slide. I came very late.
Oh, okay.
Thank you.
Okay. So then the other one has to do with the use of, so Prof, my former DDG. The good thing is that this particular work that we are doing, that's why I talked about the unintended effect. The moment we have the gene inside Songotra, and Songotra is not a nitrogen use efficient variety. If our variety becomes nitrogen use efficient, then there is a problem, which means that we have to stop the work at that particular point, because the aim of the transformation was to confer resistance against Maruca, which is a key pest of the crop. So if even a different insect feeds on it and dies, then there's a problem somewhere. That means what we call the unintended consequences has happened. So we have to stop the work at that particular point. So because of that, it is not possible for the cowpea material that we have developed to be, its nitrogen use to be different from the non-transgenic one. But if we introgress the gene into a variety that has high nitrogen use efficiency, then we expect it to also possess that particular character or trait. So this trait is just against the insect and not against any other thing. Yeah, not against any other agronomic trait. So it is not actually efficient in using nitrogen. It has to just be like the conventional variety. Then the farmer trials that we did, it was actually done over 2 seasons. So the same farmers were brought in 2 different seasons to actually conduct those evaluations. Then I think the last question was, is the BT better than what is so wonderful about the BT1? What is so wonderful about the BT material and the reason for which we are actually pushing for it is simple. It reduces the number of insecticide sprays. So I remember at one of our meetings with one of the ministers, I was saying that, you see, when we spray, we end up, our farmers, they always tell us that the wearing of the personal protective equipment is quite uncomfortable for them because they sweat a lot in it. So you see a farmer wearing a t-shirt and wearing slippers and shorts and spraying. And when they do that, the effect is that sometimes they have all kinds of burns on their body. There's even been instances where a farmer, after spraying, went home to eat without even washing down, and the farmer collapsed. They blamed it on the second wife, but later on, investigation showed that it was actually the pesticide that was applied that caused that kind of pesticide poisoning. So these are real issues we have in our farming communities. The cost of the chemicals, if a farmer is to spray 1 liter, which is about, let's say currently about 60, 70 cedis every 3 days, and the crop has a lifespan of about 2 months, that's 60 days in a year, and you are doing that almost every 3 days, you can imagine the cost as compared to just spending 140 Ghana cedi and you are done. Then the repeated spray, myself and you may not be cowpea farmers, but then we'll go and buy the grains from the market to feed on. If it was harvested shortly after spray, you could have pesticide-related poisoning after consuming any meal made from it. And I remember a couple of years back in Savulugu, there was a family that fed on pesticide-contaminated cowpea because they had run short of food. And the entire family got poisoned. They nearly lost their life just from that pesticide poisoning for the timely intervention of health personnel in the district. So these are realities, and that is what is making the BT cowpea wonderful. We are saving the cost of production for farmers. We are safeguarding the health of consumers and farmers. And also we are trying to make farmers better off because low cost of production, high yields means that financially it is more rewarding than the conventional variety. Then Prof was asking whether we didn't test it against pre and post emergence fungal disease and all those things. So like I answered to the earlier question, if the variety we are using is susceptible to a disease, we don't expect it to be resistant to that disease because the trait of interest is not to protect it from the disease. So the technology is such that the moment you are targeting, if you are targeting A and you start seeing B, then you have to stop because that is not what, that means there's an unintended effect. And once you cannot account for that, you don't know the consequences beyond that particular point. So Songotra, as far as we know, is only susceptible to macrophomina. And that is why we are talking of other varieties that we are trying to get the gene eventually into, such as Kekhouse Benga, Wankai, Apabala. And with this, a farmer that is cultivating in an area that is that has a lot of macrophomina could choose to go for Kekhouse Benga instead of Songotra. Then another farmer who is cultivating in a place that macrophomina is not a problem could choose to grow Songotra. So these are the kind of things that we are doing just to give farmers a wide range of options as far as this BT cowpea is concerned. Thank you very much. I hope I've answered the questions.
So, fellows, the floor is still open for your comments and your questions.
Please, did you answer the question on usage of fertilizer that Mama asked? Maybe I missed that one.
Yes, please, I did.
Okay, all right, thank you.
Yeah, I was saying that the gene targets only the insect. So if the variety is not nitrogen use efficient, then when we have it in there, it will not make it nitrogen use efficient.
Oh, then how about if now by using it, it doesn't use any what do you call it, fertilizer at all, whether you apply fertilizer or not? Or on the other hand, if it tends to use more fertilizer, what would you say? Then you will stop the experiment?
No. So what happens is that if you have the material, you go through the normal agronomic practices as far as cowpea production is concerned. That is how it works. So if as part of your cowpea production, you want to apply fertilizer, you can still go ahead to do that. But then using this BT variety will not make it such that you don't have to use fertilizer, and doesn't use more fertilizer than the conventional variety. It's just supposed to be like the non-transformed material, but the only difference is that it protects itself against damage by the insect of interest. That's all.
Do you have anything to add to what Jerry has said concerning the issues that have been raised? Okay, fellows and colleagues, I would like to, it looks like we don't have any more issues. But Jerry, I think Prof. Aite mentioned that if you can come up with a summarized form of the advantages of the technology, such that we can maybe send it to all the fellows, so on their own they can go through. I think it will be summarized form. Are you promising us? The two, yeah. And I'll make sure that you do it.
Will be useful to have this for people to know exactly, to understand the process, because it is a lack of understanding of the process which is here. Yes. Thank you.
So, fellows, I would like to thank you very much for spending time with us. And Jerry is around. They have promised that they will give us a summarized form of the process, so that as members of the academy, we can also be ambassadors of this new technology that is coming to help our farmers reduce the number of times they spray and even the effect of the spray on their bodies. Jerry and OFAP, on behalf of the Food and Agri chapter, and on behalf of the president of the academy, I would like to thank you very much for organizing this event for us. You have promised us that you're going to make available to the academy a summarized form of the process that led to this event. And we'll look forward to having it. On this note, we'll draw the curtain on today's meeting, and we want to thank Jerry so much for traveling all the way from Tamale to come and educate us. It's a process, and if any fellow has any question, you can drop it either to me, and I will forward it to Jerry for him to respond appropriately.
So we thank all of you for your time, and especially the question that came up. This is exactly what we want to do. So like Prof raised, the understanding will go to all of us. It's a matter of choice. You can decide to go for it or not to go for it. But the scientist is only interested in letting us know what is good in the new seed or the new technology. So I want to say that this program has been organized by OFAP Ghana. OFAP Ghana was launched in Ghana in 18th August 2011. The main mandate is to make sure that such scientific information on biotechnology, GMOs, and gene editing and the rest, which are new to us or relatively new, will reach a wider community. And this is what we stand for. And then OFAP is actually hosted by CSIR head office. So if you need any information, you can always look for OFAP in that building, the CSIR head office building. And this is one way by which we try to share the information or knowledge on these new technologies. We do that through conferences, workshops, and other stakeholder meetings. What we do is not to one preferred group, but any group that is interested in asking OFAP to share such knowledge with them, we are always ready. We can negotiate and let you know when we can do and what we can offer. So this is the little that I want to add here, that OFAP is always ready to support with knowledge on the new technology. Thank you all for coming.
I went to give a lecture at Prempeh College sometime. I went to Prempeh College myself. And when I entered, because I was going to talk about GMOs, I mean, you could see the atmosphere was so charged. But I took my time to walk them through the pros and cons. And at the end of the day, they embraced the technology. They said, why is it that we have allowed people to, misinform them? So the ball is in your court now. The technology is safe. The technology works, but people are against it. Let us try and demystify this technology. It's a safer technology. But of course, people have the right to, like you said, so we are making choices available so that farmers can decide either to go with their traditional varieties or this one that will give them more yield. Thank you very much once again, all of you.
I just want to say that it's actually a promise. So we are going to do it because we work with the media, and the media understands the language of the people. If we as scientists will speak science, they will speak the language of the people. So we're going to work with them to make sure that each and every one, not even non-scientists, would understand what is going on.